Dear friends, let us talk about the identification of fossils and codes of systematic nomenclature. Fossils in a rock are characterized by some peculiar features which enable it to be placed in a taxonomic framework, and that is what is meant by its identification, obviously. And there may be a variation of the precision to which the fossils are identified. However, a species designation is usually unnecessary. It may, for example, be possible to establish a marine origin for sediments simply by noting the presence of fossil remains of cephalopoda, which is a class, or echinodermata, which is a phylum. Paleoecologic analysis given in a well understood stratigraphic framework seldom requires a species identification. Similarly, in stratigraphy, it may be possible to distinguish between identical rock units of differing age by fossil identification of phylum, class, or order. In establishing a classification, lower taxonomy categories are generally delineated first and then assigned to higher categories. The procedure is normally reversed in identification. Suppose a fossil is covered with polygonal plate-like elements arranged readily. Columns of plates extend from one pole of the roughly spherical skeleton to the other. Most plates have one or more knob-like structures called as tubercles, which, by analogy to the living relative, are points of attachment for movable spines. Most plates have one or more knob-like structures called as tubercles, which by analogy to the living relatives are points of attachment for movable spines. This combination of the characters suggests that the specimen belongs in the phylum Echinodermata and within it. In the class Echinoidea, no other class of this phylum bears such characteristics. We thus eliminate at sight all but one class, thereby reducing the identification problem to choice among thousands of species. Phylum identification is therefore the initial step and is important to give the full working knowledge of the taxonomic characters most useful in distinguishing phyla. There are approximately 12 main phyla in the fossil record, and each is defined by an average 6 to 10 characters. This entry discusses the evidence for speciation and the patterns of species multiplication and its association morphological change as revealed in the fossil record. Speciation is regarded as the key stage in the generation of biological diversity, as it is the point at which reproductive isolation is achieved. The hallmark of the biological species, many thousands or many studies of modern species concerns themselves with the short-term genetic fluctuations between population of a single species. How those fluctuations may come out and how they are maintained. Such studies are not possible in the fossil record. On the other hand, the fossil record documents extended time scales and may therefore identify patterns of long-term changes relevant to the different theories of speciation that are unavailable in the recent world. Irrespective of the modes of speciation, the fossil record demonstrates repeated instances, geologically speaking, of the sudden increases in the number of species, and these are usually associated with rapid morphological divergence. Such events are commonly referred to as radiations, more usually adaptive radiations, and are inferred to have come about by causes intrinsic to the organisms and or extrinsic causes. Although studies of radiations involving populations and closely related species are studied in the recent world, broad-scale patterns can only be studied through the time dimension supplied by the fossil record. Species recognition and speciation in the fossil record. There is a plethora of species definitions and criteria for recognizing species in the recent world, but few are applicable to the fossil record, where only a limited amount of morphological data, usually the skeleton, is available for study. The biological species concept of Ernst Mayr is probably the most favored by people studying the recent world. Species status is achieved when populations diverge genetically to such an extent that the reproductive cohesion is broken. There may be several reasons for the fragmentation of reproductive continuity. Geography, ecology, and behavior are most commonly cited. Although Mayr stressed geographical isolation, the parent species may continue to live alongside the daughter species. In practice, most modern species are not recognized on reproductive criteria. Instead, some measures of the morphological, genetic, behavioral, or ecological difference is used as a surrogate for reproductive incompatibility. As such, paleontological species recognized almost exclusively on morphological differences are equally as valid as modern species. In fact, there has been empirical justification for equating genetic differences with morphological differences between modern species of some groups, such as bryozoans and Darwin's finches. It is, however, recognized that morphological difference may not always indicate species differences. For example, sibling species or polymorphic species, including mimetic species. The evolutionary concept of George Gaylord Simpson defines a species as an ancestor-descendant sequence of populations changing through time with their own trends and tendencies. This definition embraces asexual species. Such a definition allows for one species to change gradually into another, that is the anagenesis, as well as instances where a species is budded off or where one species is split into two or more, that is the cladogenesis. The Henningen species concept, which is named after the German entomologist Wilhelm Henning, like the biological species concept, is based on the criterion of reproductive isolation. However, the species limits are recognized only at the point where one species is split into two or more and the interbreeding pattern of gene flow is disrupted, at which point the ancestral species cannot by definition live alongside the descendants. This species definition is strictly dependent upon the shape of the phylogenetic tree. Coming to the second module.
Cut.
Recognition of species. Species in the fossil record are nearly always recognized on morphological criteria. There are some exceptions, such as instances where species are distinguished from one another purely on the basis of stratigraphic occurrences or geographic location. But these are usually accepted as stopgap measures to be revised when more information about morphology becomes available. Monophyletic species are recognized on the basis of possessing one or more unique morphological characters, and this corresponds most closely within the Henningen species concept. Species viewed in this light have the same ontological status as higher taxa such as genera, families, orders, etc., In reality, very few species can be recognized in this way, and logically, it denies the existence of ancestors, since ancestors have no unique features. Typological species were once commonly recognized, whereby a single specimen that is usually the first discovered or the most complete was chosen as the Linnaean type, and this formed the nucleus of morphological variation considered worthy of a species status. How much variation was to be allowed before a new species was recognized was never stated. Such species are usually frowned upon now as being steeped in the philosophy of pre-Darwinian essentialism. Yet, there remains many instances where such species are still used. Phanerotic species arose out of the numeric taxonomy. Here, species are measured for as many morphologically continuous variables as possible. The variables are then subjected to multivariate analysis and clusters. These species are then recognized, although this may appear to be objective and theory-free. It is heavily dependent upon the original samples analyzed and the multivariate analysis algorithms used. It is not always easy to identify which of the individual variables is contributing to the differences, and therefore it is difficult to diagnose the species. Phylogenetic species are the smallest diagnosable assemblages of the specimens, showing a unique combination of characters. Combination is the operative word here, since there is no requirement for identifying unique characters. For example, monophyletic species recognition. Of all the recognition criteria, this most faithfully agrees with all of the process-based definitions of a species, as well as agreeing with the day-to-day practice of paleontologists. The estimates of species definition longevity may depend on which of the above ways the species have been recognized. Surveys across various fossil groups give average figures that range from million years for small mammals, where generation time is very short, to 13 million years for dinoflagellates, which are largely asexual. Speciation in the fossil record, it is generally accepted that most speciation events, within the possible exception of those caused by the hybridization and polyploidy, that is chromosomal duplication, are too protracted to be detected in the recent world and too brief to be seen in the fossil record. The lower limits of the time that can be successively sampled in the fossil record appear to be in the order of 5,000 to 10,000 years, and such deposits are very rare. Restricted to the lake deposits of relatively short duration and small geographic range, and some deep sea deposits. Nevertheless, there have been many studies seeking to establish the precise pattern of speciation. There are two polarized theories on the tempo and pattern of speciation, punctuated equilibria and phyletic gradualism. Both have support from the theoretical and empirical studies in the modern world, and it is likely that both modes of speciation can happen. Phyletic gradualism holds that morphological evolution is gradual and occurs independently of speciation. Most of the studies supporting this mode of evolution involve organisms that can be minutely sampled through continuous rock sequences. Some gastropod species shows an example of changing or replacing another in ancestor-descendant sequence, that is gradual speciation by anagenesis, by a shift in the mode or the modal shell from as measured by morphometric discriminant analysis. In this example, the gradual change has been correlated with increasing depth of water. The populations intermediate between the two species were presumed to have lasted for 73,000 to 250,000 years. In this pattern, there is no rapid shift in the morphology, and in studies of this kind, the species limits are usually recognized by comparing the morphological variation over a period of time with morphological variation of modern species and dividing up the continuum accordingly. In another study involving radiolarians recovered from Pacific Ocean deep-sea drilling cores, an instance of speciation was detected occurring during a period of about 500,000 years. In this case, both the ancestral and daughter species showed marked deviation in many aspects of the skeleton form. Further, it is interesting to note that morphological deviation, although slightly accelerated during the speciation event, continued for some time after the separation of morphotypes, suggesting strongly that morphological evolution is decoupled from speciation. In this instance, both parental and daughter species showed morphological evolution, and it may have been possible to suggest that a single ancestral species gave rise to two daughter species. The author of this study refrained from this because the ancestral species which still lives today show a high degree of polymorphism and geographic variation, the scale of which is comparable to the variations seen in the lineages in this study. It needs to be pointed out that many, but not all, of the studies that support phyletic gradualism report information on the continuously varying morphologies such as lengths and shapes. These parameters are inherently gradualist in their measurement, unlike many characters used in cladistic analysis, such as presence or absence of 4 toes versus 5 toes, in which there can be no intermediate, the taxonomy and the concept of nomenclature. Taxonomy is a fundamental and dynamic science dedicated to exploring the cause of the relationships and similarities among organisms. Classifications are theories about the basis of natural order to identify a fossil form correctly, and indeed are keys to find out further information about it. Sound classification and nomenclature lie at the root of all biological and paleontological work. Without them, no coherent and ordered system of data storage and retrieval is possible. Taxonomy, or systematics as it is sometimes known, is the science of classification of organisms and is the oldest of all biological disciplines. In the formal nomenclature of any species, living or fossil, taxonomists follow the biological system of Linnaeus, whereby each species is defined by 2 names, the generic and specific or trivial names. For instance, all cats, large and small, are related, and one particular group has been placed in the genus Felis. Of the various species of Felis, the specific names of Felis catus, Felis leo, and Felis pados formally refer to the domestic cat, lion, and leopard respectively. In full taxonomic nomenclature, the author's name and the date of publication are given after species. For example, Felis catus, which in bracket will be written as Linnaeus 1778. In paleontology, it can never be known for sure whether a population with a particular morphology was reproductively isolated or not. Hence, the definition of a species in paleontology, as in most living specimens, must be based almost entirely on morphological criteria. Moreover, only the hard parts of the fossil animal are preserved and much useful data has vanished. A careful examination and documentation of all the anatomical features of the fossils has to be the main guide in establishing that one species is different than the related species. In rare cases, this can be supplemented by a comparison of the chemistry of the shell, as has proved especially useful in the erection of higher taxonomic categories. Within any interbreeding population, there is usually quite a spread of the morphological variation. On a broader scale, there may be both geographical and stratigraphical variations, and all these must be carefully documented if the species is to be ideally established. Such studies may be very useful in evolutionary paleoecology and paleontology. When a paleontologist is attempting to distinguish the species in a newly discovered fauna, say of fossil brachiopods, she or he has to separate the individual fossils out into groups of morphologically similar individuals. There may be 8 such groups, each distinguished by a particular set of characters. Some of these groups may be clearly distinct from one another. In others, distinction may be considerably less, increasing the risk of greatly or greater subjectivity. These groups are provisionally considered as species which must then be identified. This is done by consulting paleontological monographs or papers containing detailed technical descriptions and illustrations of previously described brachiopod faunas of similar age, and comparing species point by point. Some of these species may prove to be identical with already described species or show only minor variation of a kind that would be expected in a local variant within the same species. Other species in the fauna may be new, and if so, a full technical description with illustration must be prepared for each new species, which should be published in a paleontological journal or monograph. The description is based upon the type specimens, which are always thereafter kept in a museum. Usually one of these, the holotype, is selected as the reference specimen, and fully illustrated comparative detail may be added from other specimens called paratypes. There are various other kinds of specimens. For example, a new type may be erected when a holotype has been lost, or when a species is being redescribed in fuller and more up-to-date terms. When no type specimen has previously been designated, a new genus will be designated as Gen Nov by its author, this following the generic name, a new species as SP Nov and new subspecies as SSP Nov. The new species must be named and allocated to an existing genus, or if there is no described genus to which it pertains, then new genus must also be erected. Taxonomical hierarchy. Although all taxonomic categories above the level are to some extent artificial and subjective, ideally they should as far as possible reflect evolutionary relationships. Similar species are grouped in genera, that is singular genus, genera in families, families in orders, orders in classes, and classes in the largest division of the animal kingdom, phyla, which in singular called as phylum. There may be various subdivisions of these categories, for example superfamilies and suborders, etc., and in certain groups there is even a case of erecting superphyla. There are only about 30 phyla in the animal kingdom, and only about a dozen of these, for example mollusca and brachiopoda, leave any fossil remains. The basic principle is that morphological and other similarities reflect phylogenetic affinity, which is called as homology. This is always so unless for other reasons. Similarly, similarity results from convergent evolution, but in assessing similarities, how does one decide that which characters are important? how should they be chosen to minimize subjectivity and produce natural order groups? There is no universally accepted method of facilitating these ends, and taxonomists has used different methods. In recent years, three sharply contrasting schools have emerged. These schools of number 1, evolutionary taxonomy, number 2, numerical taxonomy, and number 3, cladism. In erecting a hierarchical classification, taxonomists use traditional and very flexible combination of criteria. First, there is morphological or phenetic resemblance, the extent to which the organisms resemble one another. Second, phylogenetic relationships are, along with phenetic resemblance, considered important. By this is meant the way, as far as can be determined, in which the organisms are actually related to each other. That is, in terms of recency of common origin, which of course grades into the evolutionary taxonomy. The order of succession in the rock record and geographical distribution may play an important part in deciding relationships. This practical approach to taxonomy, which took all the factors into consideration, has for a long time been the backbone of paleontological classification and is still considered to be the best method by stratigraphic paleontologists who place much emphasis on time in seeking ancestor-descendant relationships. For some taxonomists, however, the uncertainties and subjectivity which are almost inescapable in any kind of classification seem to be particularly acute in a classical taxonomy, as did the limitations of the fossil record in terms of preservation. Numerical taxonomy has been found very useful in some instances, but subjectivity cannot be eliminated since the operator has to choose how best to analyze the measurements made and may need to rate them, giving a few characters more importance in order to obtain meaningful results. Hence, the objectivity of numerical taxonomy is less than it might appear. The third school relies upon the phylogenetic criteria alone, emphasizing that features shared by organisms manifest in nature a hierarchical pattern evident in the distribution of characters shared amongst organisms. It is known as the cladism or phylogenetic systematics. Thus, in closely related groups, we would say shared derived characters or synapomorphies, which would be distinguished or which would distinguish this group from others. The central concept of Henning, 1966, was that in any group, characters are either primitive, that is symplesiomorphic, or derived. Thus, all vertebrates have backbones. The possession of a backbone is a primitive character for all vertebrates, is of course a derived character as compared to invertebrates, synapomorphy and symplesiomorphy, thus delineate the relative status of particular characters with respect to a particular problem. Henning endeavored to provide an objective methodology for determining recency of common origin in related taxa based upon primitive and derived characters. Such relationships are expressed in a cladogram in which dichotomous branching points are arranged in nested hierarchies. Suppose taxa A and B share a unique common ancestor. They are said to be sister groups. They both share an evolutionary novelty or synapomorphy not possessed by a taxon C. Now, C is the sister group of the combined taxa A and B, and D is the sister group of the combined taxa A, B, and C. In performing a cladistic analysis, therefore, a taxonomist assumes that dichotomous splitting has occurred in each lineage and complies an unweighted character data matrix. The more characters and character states they are available the larger the database, and larger databases are often routinely processed. How can we distinguish symplesiomorphic shared primitive character states? The most useful way is out-group comparison. Here, an in-group of which the relationships are under investigation is specifically designated and compared with a closely related out-group. Any character present in a variable state in the in-group must be plesiomorphic if it is also found in the out-group. Likewise, apomorphic characters are only present in the in-group. Henning distinguished three kinds of cladistic groupings. Monophyletic groups contain the common ancestors and all of its descendants. Paraphyletic groups are described or descendant from a common ancestor, usually now extinct and known as the stem group, but do not include all descendants. Polyphyletic groups, on the other hand, are the result of convergent evolution. The representatives are descended from different ancestors, and hence, although these may look superficially similar, any polyphyletic group comprising them is artificial. A cladogram is not an evolutionary tree, it is an analysis of the relationships. As such, it is a valuable and rigorous way of working out and showing graphically how organisms are related, and it forces taxonomists to be explicit about patterns and groups. Now, the methodology of cladistics is especially good when dealing with discrete groups with large morphological and stratigraphical gaps, and to these it brings the potential for real objectivity. So, where does that leave the potential contribution of stratigraphy in reconstructing phylogeny? Whilst a few transformed cladists negate the value of the fossil altogether, the successive appearance of taxa in stratigraphy cannot be denied as an essential source of data, however imperfect the total record actually is. Very commonly, there is an excellent correspondence between the cladogram and the rock record. On the other hand, the combined cladistics and biostratigraphic approach may throw up an unexpected pattern. The ultimate problem, not only for cladism but for all the taxonomic methodology, results from convergent evolution. Resemblances in characters or character states may have nothing to do with the recency of common origin, but from convergence, and it may not always be possible to disentangle the results of the two. Even so, cladist methodology coupled with the biostratigraphy seems to be the best way forward and an essential prerequisite for drawing up the meaningful phylogenetic trees. Thank you very much.